As I said, the fossils that we find when we dig into the ground, they turn up in the order in which we would predict from the universal tree. What do I mean by that? Well, first of all, this is the universal tree, a tree representing most life forms on the planet that we have today. I've indicated just a few of the major groups. We have different mammals here, we have various reptiles, we have insects and millipedes and crustaceans, we have some fungi, and we have some plants and then bacteria there. And they all go back to some original ancestor that, as I said, lived long ago. Now, the fact that different groups of animals are more or less related on this tree actually makes predictions about the sorts of fossils that we would expect to find. So for instance, if we look at the tree, we can see that birds in this tree has as their most adjacent, their closest relative, the group of crocodiles. Birds are closely related to crocodiles, which again are related to other reptiles. So what does that tell us? Well, this suggests to us that there must be some ancestor here that has given rise to both the crocodiles and the birds, and we should therefore be able to find a series of transitional fossils that are sort of intermediate between birds and reptiles, and moreover they should be located in the geological strata that corresponds to this, meaning we should expect to find fossils appearing after the first reptile appeared and before the first bird appeared that are transitional in form. And that is exactly what we find. We have a great deal of dinosaur to bird transitional fossils. I've listed a whole range of names here, and these are found in exactly the geological strata I suggested, namely strata located after the appearance of reptiles and before the appearance of birds. Archaeopteryx here, one very famous example, originally discovered in 1861, 2 years after the publication of On the Origin of Species, actually. This is clearly reptile-like in several traits, such as that it has fingers, teeth, and a bony tail, and it's clearly bird-like in the very important feature that it actually has feathers and it has been able to fly. But as I say, there's a whole range of other intermediate forms, and we now also have from China in particular a whole range of feathered dinosaurs that were not able to fly, but where they apparently used feathers, for instance, for insulation. Let's take a few other examples of transitional fossils. So if we look again at the tree here, we can see that the group of mammals here and the group of reptiles are closely related. This again suggests that there must be some ancestral forms that are intermediate between mammals and reptiles, and where there's been a gradual transition from the mammal to the reptile form. And moreover, that we should find these fossils in those geological strata that are after the first reptile form and before the first mammal form. And again, that's exactly what we find when we dig into the ground. We actually have a very complete set of transitional fossils, starting out with very reptile-like squatting animals, like the one I've shown on the top here, going over some of these still very reptile-like animals with sails that many of you probably had as plastic toys when you were kids, over progressively more mammal-like animals until we end up in the lower corner here, where we have animals that actually have fur and other mammalian characteristics. And again, as I say, we find these fossils in the order in which we would predict where there's a gradual change towards more and more mammal-like forms. As a special example, actually, of this transition from the reptile to the mammal form, we have some very interesting data from the evolution of inner ear bones in mammals. It turns out that reptiles have a number of bones in their lower jaw. In particular, they have two bones called the articular and the quadrate that are not present in mammals. Mammals have just a single bone in their lower jaw. However, by looking at fetal development, it turns out that the two bones that eventually evolve into the quadrate and the articular in a reptile fetus, if you look at the corresponding bones in a mammal fetus, those two same bones will eventually evolve into two bones in the inner ear called the malleus and the incus, the hammer and the anvil. bones that help mammals here quite well as mammals actually do. This, it turns out, we can also find in the fossil record. We found quite a complete set of intermediate transitional fossils going from very early reptile-like animals having both of these bones in the jaw over, and then we have a long series of progressive intermediates where these bones shrink in size and gradually move into the inner ear until the early mammals, where they were entirely located in the inner ear, the middle ear, as we have them here. Let's take one final example of transitional fossils. If we look inside the group of mammals, we can see that the nearest relative of whales is in this case cows. But more generally, whales are in fact mammals. So this would indicate that these sea-living organisms, the whales, have in fact evolved from land-dwelling organisms that are mammals. And it moreover predicts again that we should find these transitional fossils in the particular geological strata appearing after the first mammal and before the first whale. And again, that's exactly what we find. We now have a very nice range of intermediate stages going from almost completely land-dwelling whale-like organisms to more and more sea-dwelling versions with gradual loss of hind limbs as indicated on this slide. And again, I've mentioned just a few of the transitional forms that we now have. Another piece of evidence that again suggests or that supports the idea that all life is related and has evolved from a common ancestor is that of vestigial structures. A vestigial structure is defined, as I've said here, as a reduced and rudimentary structure compared to the same complex structure in other organisms. And what do I mean by that? Well, take a look at the ostrich here, for instance. An ostrich is quite clearly a bird. It has it has feathers, it has wings, but it's not able to fly. Therefore, it has vestigial, what we would call vestigial wings. The wings are clearly wings, they have feathers and look exactly like wings in other birds, but they are not able to function as wings. That's a typical example of a vestigial structure. The ostrich does use its wings for various things. It uses it for balance and for courtship and for various other things, but it's not using them for what they were, if you might say, designed, namely for flying. Now, the easiest the simplest explanation for why such a complex structure is present in an animal here, yet not being used for what it clearly ought to be used for, is that the animal inherited it from some ancestor that had a functional form. So the simplest explanation for why ostriches have wings is that they are descendants. They have evolved from previous populations of birds that didn't in fact have wings and that were able to fly. And there are many other examples of vestigial structures like this. One example is beetles. Most beetles have wings. They have wing covers that they can flip apart, and then underneath they have wings that they can fold out and fly around. However, there's a rather large group of flightless beetles which do not have wing covers that can flip apart and that therefore do not fly. They mostly live on the ground, for instance. However, many of those species, like the one I've shown here, for instance, if you take a scalpel, cut up the wing covers, you will actually find more or less fully formed wings underneath. They are just wings that will never be used to fly. What's the simplest explanation for that observation? Well, the simplest explanation again must be that those beetles inherited their wings from some ancestor who had wings and actually used them, but for some reason it has now been advantageous for it to, or at least it's not an advantage for it to fly, therefore it's lost the ability to do so. The wing covers are fused, and if we wait another few million years, it might be that they won't even have wings underneath the wing covers. Very similar example, the Texas blind salamander here. This is a salamander that lives in caves. There's absolutely no light. It doesn't have eyes, but if you again take a scalpel and cut the skin where the eyes were supposed to be, you can actually find more or less fully formed eyes underneath with retinas and lenses, etc., etc. Again, the simplest explanation for that is that these salamander evolved from some previous population of salamanders that were able to see, but since this particular salamander took up a life form where it lives in total darkness, it no longer, there's no selection for it being able to see, and if we wait again a couple of million years, it might be that they wouldn't have any eyes even underneath the skin. Final example of a vestigial structure is that of whales. Whales, as I just said, evolved from land-dwelling organisms, and they now live entirely in the sea. Nevertheless, some whale types have vestigial pelvises, even with vestigial hind limbs attached to them, very small femurs, for instance, such as the baleen whale I've shown you here. And again, the simplest explanation for that is, of course, that the whale, as I suggested, evolved from a previous population of animals that were able to walk. Going to the molecular level, there's also a great deal of evidence in addition to what I already mentioned. One of the more interesting types of evidence is that of endogenous retroviruses, or ERVs. An endogenous retrovirus, first, a retrovirus is a sort of virus that infects a cell. It has an RNA genome. It then uses a protein, an enzyme called reverse transcriptase, to convert the RNA into DNA, and that DNA is then inserted into the genome of the host organism. HIV is one example of a retrovirus, and this is one of the reasons why it's so difficult to get rid of HIV, is that it actually inserts its genome into the T cells, and it gets replicated along with the T cell when that copies itself. Now, if such a retrovirus infects a cell in the germline, a cell that will eventually form a sperm or an egg, then it might be that that inserted piece of retroviral DNA is transmitted to the next generation, if that particular egg or sperm is fortunate enough to actually be part of the next generation of animals that grows up. And as it turns out, there are actually quite a lot, and we will then call it an endogenous retrovirus. That retrovirus is present then in the next animal growing up. Typically, the retrovirus will at this point have mutated, maybe lost a part, or maybe had point mutations, so it's no longer functional. But it will be in the genome and be clearly recognizable as a retrovirus. Such endogenous retroviruses, as we call them, are actually fairly normal in the human genome, for instance. They make up about 5 to 8% of the human genome, with about an estimated 100,000 fragments of ERVs in the human genome. Now, the interesting thing is that a lot of these endogenous retroviruses are shared between humans and other animals. For instance, we share almost all our endogenous retroviruses with chimpanzees. Of the 100,000 ERVs that we have, maybe only a few hundred are specific to us, and similarly, only a few hundred are specific to chimps. The rest, the vast majority of these 100,000 endogenous retroviruses are shared between us and chimps, in the sense that we have the same endogenous retrovirus sequence, more or less, in the same location in the genome. We also share endogenous retroviruses with gorillas. We just share a bit less than we share with chimps. We also share endogenous retroviruses with orangutans, but again, we share a bit fewer with them than we do with gorillas, and even fewer than we do with chimps. And that goes on all the way down to gibbons, Old World monkeys, and New World monkeys. Now, the easiest explanation again for why this, that means that we actually again see this pattern of a nested hierarchy of sharing, okay? So we share some of our endogenous retroviruses with chimps. We share some with gorillas, and some of the ones we share with gorillas we also share with chimps. We share some with orangutans, and only some of those we also share with gorillas, some of those we also share with chimps. So we have, again, a nested hierarchy of traits. The simplest explanation for that is, again, that we have inherited these endogenous retroviruses from a previous ancestor, and that we have evolved from some common ancestor. For instance, the retroviruses, the endogenous retroviruses we share with gorillas, have probably inserted into the genome of the common ancestor we have with gorillas, lived maybe 8 to 10 million years ago. At some point, endogenous retroviruses were inserted, and all the species that have evolved from that original population now share all of those retroviruses. Generally, if a retrovirus inserts into some ancestor at some point, then all the species to the right of that insertion point will have those retroviruses. And that's exactly the pattern we see, and it's most easily explained by common descent, by us having evolved from a previous common ancestor. Another piece of molecular evidence that is quite interesting is that of the chromosomes in humans versus the rest of the apes. It turns out that all the great apes, except for humans, have 24 pairs of chromosomes. They have 48 chromosomes. Humans have 23 pairs of chromosomes. We have 46 chromosomes. Interestingly, if we look at human chromosome 2, which I've tried to show in cartoon form here, then it turns out that the sequences that we have on human chromosome 2, we can find almost identical sequence in the chimpanzee genome, but it's not located on a single genome, a single chromosome, sorry. It's located on two separate chromosomes that we now call 2A and 2B. So the simplest explanation for why this is so is that in some ancestor, the ancestor of all apes, there were these two separate chromosomes. In the ancestor of humans, these two chromosomes appear to have fused more or less end to end, forming one long chromosome from the two original chromosomes. And this can be seen in such things as banding pattern of the human chromosome 2, which fits to each of the separate chromosomes in chimps and gorillas, for instance. It can also be seen in the fact that we have what's called telomeres. Most chromosomes in eukaryotic organisms have structures called telomeres at the end of their chromosomes, and other structures called centromeres at the middle of the chromosomes. And what we can see in the human chromosome 2 is that we actually have some residual parts of some ancestral telomere sequence in the middle of human chromosome 2, and we also have remnants of an extra centromere. And that's exactly what we would predict if chromosome 2 evolved by fusing two original chromosomes. So a very interesting piece of evidence there. A final piece of molecular data supporting this fact of evolution, this idea of common descent, is that of the biosynthesis of vitamin C, or ascorbic acid. It turns out that practically all animals are able to synthesize vitamin C from a molecule called D-glucose. I've sketched the pathway up here. It's a number of enzymatic steps where step by step the glucose molecule is converted into ascorbic acid, or vitamin C. Humans and other primates, it turns out, are not able to synthesize vitamin C, and that's why we need to have a dietary intake of the vitamin in order to avoid scurvy. The reason that we are unable to synthesize vitamin C, it turns out, is that the final enzyme in this pathway is defective or is missing in humans and these other primates. This enzyme is called GULO for L-gulonolactone oxidase. So as it turns out, the reason that we don't have a working GULO enzyme is in fact an interesting one, because if we look carefully in the genome of human beings, if we look in the DNA, we can actually find a remnant of a functional GULO enzyme. So here's an alignment of part of exon 1, part of the one of the exons in the GULO gene from rat and from human. Rat has a functioning GULO enzyme. It is able to synthesize vitamin C. Humans do not. However, we can see from this alignment that we can actually find the DNA sequence that resembles to a very high degree, that in the human genome we can find a DNA sequence that resembles to a very high degree the GULO gene that's present in rat. There's a few point mutations, but on the whole it has a very similar DNA and in fact also amino acid sequence. However, there's a number of changes, including, for instance, changes such as this single nucleotide deletion, which will cause what's called a frameshift mutation, rendering the rest of the protein non-functional. It actually also turns out that the human gene here lacks some of the exons that are in, for instance, the rat, and that is another reason that this gene does not work. But the main point here is that there is, in fact, in the human genome, a relic of a functional GULO enzyme. What is the reason? Again, the simplest explanation must be that we have, of course, inherited this gene from a common ancestor that lived many millions of years ago. At some point it mutated, maybe because we were living in an ecological niche where we had plenty of citrus. Therefore, there was no selective pressure to maintain the gene, and these mutations, therefore, were not selected against. But the bottom line is, the simplest explanation is, we have had a common ancestor with the rest of these animals. We have inherited the gene from that common ancestor. And I could go on and on. I'm just going to leave the last slide here with many of the other sorts of observations that are telling us that common descent is in fact true, that there has been a common ancestor many millions of years ago, billions of years ago, and that all life on the planet has evolved from that ancestor.